Dr. Jimenez's microbiology group.
And my name is Mina. So today we're just going to present you the antimicrobial research project we're working on since the beginning of summer. So as you know, bacteria have become antibiotic resistant over the years due to us consuming antibiotics on a daily basis in our food and so many other factors. And so, for example, penicillin, which is a very known antibiotic, which used to cure so many diseases a couple years ago, is no longer useful now. So in our project, we studied 14 samples of Staph aureus and pertaining its
Its reactivity to various antibiotics,
pharmaceutical antibiotics and herbal remedies. So funny thing we found that, for example, lavender oil and tea tree oil perform comparably to certain antibiotics we were working on. So in the future, we will study more types of infections causing bacteria and also more herbal remedies.
So these were the various methods that we used to develop our project. So this one, of course, is where we transferred various samples of Staph aureus to agar plates, and we placed discs that had different antimicrobial substances on them. So some had antibiotics, some had the oil that they were soaked in. We incubated them overnight, and then the next day we ended up measuring the inhibition ring around them, which basically means the bacteria decided not to grow there because it hurts them. So this was the DNA prep kit that we used. We basically have to take the bacteria and dissolve it into DNA pretty much. We use various buffers and washers and stuff like that to kind of extract the DNA from it so we can process it in other machines that we need to use. PCR, polymerase chain reaction. So we took the prep DNA that we used, that we made, and we mixed it with primers. So the primers are basically the specific strand of DNA that we want copied in order for us to examine it further and determine whether that bacteria is resistant to certain antibiotics or not. So you put it into the machine, and as we have like various settings here, I don't know if you guys can see, but it's all different temperatures. So the extremely high temperatures are where the DNA strand gets ripped in half. So the double helix turns into two separate ones. And as it cools, the primers can attach to different opposing sides of the DNA. And as it cools further, they replicate that specific strand. So once we were able to isolate the strands that we want and the PCR is finished, we basically stain the samples that we have with ultraviolet light, and we run it through this gel electrophoresis. So basically the DNA is negatively charged, and the other end of the gel electrophoresis machine is positively charged. So it kind of pulls it through. The smaller fragments tend to move faster, and the larger fragments tend to move smaller. Anyway, the point of the fragment is if it's in a specific, so we end up counting certain bands. If it's within a specific band of the primer that we tested, then we know it's a positive, and that specific bacteria is positive for that resistance gene.
So through all this, through PCR examination and gel electrophoresis, we found out which genes were antibiotic resistant, and so we're just gonna work our way forward from that.
We were thinking of implementing maybe food grade oils.
Yeah.
So instead of just these being topical for like maybe infections, we could potentially use them to cure infections like within us. We're also going to be testing more types of bacteria, not just the Staph aureus. So we'll be continuing. There's definitely some holes in our data sheets that we need to fill in, but at least with the data that we do have, novabiocin seemed to be the strongest antibiotic with the largest inhibition rings. Lavender oil was surprisingly comparable. Also tea tree oil. And then the samples that we were able to identify as positive for the antibiotic resistance, they tend to have smaller inhibition rings.
Yeah, any recommendations on antibiotics or bacteria, let us know, please.
Any questions?